His research line is molecular epidemiology of rabies and comparative immunology of marine mammals. Okay? So this class will be until 11:30. Okay? So Dr. Takuya Ito, can you come to the front and start the class?
Thank you. Good morning, everybody. My name is Takuya Ito from Nihon University Veterinary Research Center. Today I will be talking about rabies, especially molecular epidemiology of rabies in the world. At first, thank you very much for inviting me, and I appreciate for having this opportunity. Thank you so much. Rabies, this disease have many phases. For example, this disease is zoonosis, an animal-derived human disease, and sometimes transboundary disease, emerging disease in some region, and re-emerging disease in some region. And in developed country, the imported disease, for example, in Japan, we don't have natural rabies nowadays, but 2006, we have two cases imported rabies from Philippines. Maybe your US country have a lot of imported case from other country. And the many rabies cases occurred in tropical regions. And rabies case fatality rate is 100, almost 100%. So this is very severe lethal disease. And this disease have very long incubation time, so latent infectious disease. So many countries, like Japan and your country, this rabies disease is neglected, especially the developed country. This is a real rabies case, dog rabies in Uganda. This dog is killed by some people, and this dog attacked to 3 children before dead, and children go to hospital, and you can see the child don't have shoes and naked foot. On the other hand, the adult person has shoes and long pants. So children is very dangerous for rabid dog attack. So many cases is dog rabies victim is child. Why rabies is dangerous? Rabies affect central nervous system and causes terrible neurological symptoms. Here we watch the human rabies and animal rabies.
角膜陰圧標本などは診断的価値がある。臨床診断の方が信頼性が高いこともあります。鑑別診断は常に考えておかなければならない。
In this videotape from South Africa,
精神症状を伴った腸チフス、精神分裂病、薬物中毒、脳炎、脳症などがある。しかし、 動物に噛まれたという信頼できる起用歴があり、狂犬病に特徴的な症状、すなわち胸水発作、胸風症、意識が間欠的に生命になったり、強い不安が
Maybe he was bitten by dog 1 or 2 months ago.
人の狂犬病症例で見られる症状の多くは驚くほど動物の症状に似ている。
Human rabies symptom is resembled to the animal rabies symptoms.
は14歳以下の子供である。ゴドロントン博士の説明を受けたので、ここで実際に狂犬病の症状の詳細を見ることにしよう。
Symptom is fever, sweating, and fatigue, lethargy. Very tired and shows aggressions, and sometimes biting like a dog. And typical symptoms is aerophobia, don't like wind. This is also typical symptoms. Hydrophobia, don't like drink water. He was very thirsty, but cannot drink water, so the dehydration. He has paralysis of throat. And hallucination in the latent phase. And coma. Lastly, dead. It's a rabid dog symptom. It's very aggressive and characteristic howling, characteristic crying. Crying continuous. Sometimes the rabid dog has he has wound in the leg and attacking, chewing inanimate objects. Rabid dog has very powerful biting, so if the human being bitten by rabid dog is wound, and injury is very severe and deep, so very dangerous. And this is a typical symptom, bone-in throat symptom. It is dangerous for dog owners. And inability to swallow. He cannot drink water. So indicate dehydration. And sometimes choking and vomiting. The last stage is incoordination. I cannot stand up steadily. This is the final stage of rabies. This is cattle rabies.
脳を侵す他の疾患と混同されることが多い。喘ぎ呼吸と長く泣くことが
It's a typical crying
特
voice,
徴的な症状である。
wind suckling and blowing.
狂犬病の牛の多くは食欲が減退する上、唾液の流出が多量になるため、顕著な脱水症に陥る。
Usually it's hypersalivation and dehydration.
食欲が低下しているにもかかわらず、
It's abnormal appetite.
犬と同様に
It's also bone-in throat symptom. It's like a dog
飼い主や獣医師が閉塞物があると誤解し、これを除去しようと試みることがあるが、犬の場合と同様に感染の危険がある。
It's dangerous for the owners.
口頭、抗菌の麻痺により嚥下困難が生じる。狂犬病の牛は落ち着きを失い、怒りっぽくなり、地面に穴を掘ったり、
Sometimes
誘引がなくても
rabbit
攻撃したりす
cattle
る。
shows aggression.
協調運動障害はどの動物でも起こるが、
It's a common symptoms in coordination.
障害は後ろ足から現れて、よろめき歩行となる。
This paralysis is from leg and can't stand up.
やがて起立不能となり、嘔吐するが、四肢には走るような動きが見られる。
This is also final stage of rabies. It's goat rabies. Incessant bleating continuously. Very typical, the characteristic crying.
協調運動障害、後ろ足の脱力や麻痺も多く報告されている。ここに示した性欲行進は注目すべきものである。
Sometimes the hypersexual behavior and abdominal libido in goat rabies. Sometimes shows aggression, abnormal behavior.
すべての狂犬病動物に共通することは、確実に死に向かって進んでいくことである。狂犬病の
It's
羊
sheep
の症状は、
rabies
狂犬病の
symptoms,
牛やヤギに
lip
極めて
twitching
似
and
ている。
salivation.
多量の唾液流出と口の周囲の痙攣は草食動物に共通して見られる。しかし、狂犬病の羊の何割かは何ら症状を示さずに
But
急に死亡
sometimes
する。
sheep rabies, no symptoms and death. The pig rabies,
狂犬病の豚も猫のように攻撃性が高まり、激しく噛みつく。
rabbit
子
pig
豚は踏み殺されたり、
shows
四肢を噛み
aggression
切られること
and
も多い。
sometimes
性
killed
欲の更
piglets.
新や感覚の異常も見られる。狂犬病の馬にはまず行動の変化が見
Horse
られる。
rabies,
古い噛み傷を舐めたり噛んだり、
behavior change and
病気が進
biting.
行すれば興奮性が増し、 立ち上がったり蹴ったり
and
する
shows aggression.
咽頭後頭筋の麻痺、性欲の更新、よろめき歩行
And walking slowly.
馬は目的もなく歩き回ることと、映像の子馬のように後頭筋の麻痺による嚥下困難が主な症状となる。
This young horse shows cannot drink water and dehydration.
唾液流出と筋尿のために体力が失われて脱水症に陥る。地域における森林型
Wildlife rabies. It says mongoose. Mongoose is distributed in India, Africa, and West Island, West Indian Island, like Puerto Rico.
イエローマングースがいるところには森林型狂犬病があると考えてよい。狂犬病のイエローマングースは人や他の動物を恐れなくなり農場に接近する。
Rabbit mongoose shows a very high aggression and very important vector of rabies for livestock animals.
症状が急速に進行している。奇妙な叫び、後ろ足の麻痺、穴掘り動作、よし間もなく死亡する。
This is also final stage. Cannot stand up and go to death. This is jackal.
狂犬病のジャッカルは人や家畜などを恐れなくなる。
Jackal rabies in
狂犬
Africa.
病のジャッカルの主な犠牲動物は牛であり、水場で襲われることもある。また、
This is very interesting. This brave jackal attacked to elephant.
非常に狂犬病を伝播しやすい動物である。
Very important vector in Africa. Okay, rabies infect every mammals, including human beings, and this disease onset means death. So this disease is a lethal disease. And some wildlife animals often transmit rabies virus as vector. It is impossible to diagnose it before onset because rabies definite diagnosis needs brain sample. However, human rabies is 100% preventable through prompt appropriate medical care. Even 55,000 people die annually. This appropriate medical care is post-exposure prophylaxis. This is a recommendation by WHO. As soon as possible after the exposure, dog bite, for example, dog bite occurs, treatment consists of through wound cleansing for a minimum of 15 minutes using water, soap, and biocidal antiseptics. For example, the ordinary antiseptics, providone iodine or ethanol, followed by the administration of rabies passive immunization. Passive immunization is a gamma globulin, and purified rabies vaccine of proven efficacy. Usually 4 or 5 times vaccine shot as booster shot repeatedly. This is the infectious route of rabies. Rabies dog or rabies fox, their saliva contain many virus particles, and rabies virus penetrates into the peripheral tissue, and once rabies virus propagates in peripheral tissue, for example, muscle, virus, virus enter to nervous system and go to central nervous system, like brain. If once propagation of virus in brain go to this, but PEP post-exposure prophylaxis can interfere penetrate to the nervous system. This is interfere. So this is a good protection for rabies. We need a brain sample for definite rabies diagnosis in laboratory. This is a fox brain, and this is a bat brain, very, very tiny. This is a dog brain, suspected dog brain, and cat, and we take a part of several parts of brain tissue, and we make specimen, stamp specimen for rabies diagnosis, and react with rabies virus, anti-rabies virus antibody with fluorescence. So if the rabies positive, this is a positive reaction. It's this is a positive result affected dog brain, and this picture shows a cell line inoculated with brain sample. It is also the positive reaction. And this is a diagnosis for the bat rabies using the same procedure, fluorescent antibody test. This is a positive reaction. This photograph, I took this photograph, this sample derived from rabid cattle in Brazil. Another diagnosis technique is rabies virus isolation by mouse inoculation test. We make emulsion of the suspected rabies animal brain and centrifugation and taking the sample from the supernatant and inoculate to the suckling mouse or juvenile mouse brain and observed clinical symptom for 2 weeks or 3 weeks. And if we find clinical symptom in this mice, we remove the brain sample and fluorescent antibody test again to confirm rabies infection. This is another rabies virus isolation method by cell culture. This technique use cell line, very susceptible, rabies virus susceptible cell line instead of mice. This is a very strong positive reaction by fluorescent antibody test. Here, I would like to introduce rabies virus scientifically. This virus classified mononegavirus, and family is Rhabdoviridae. Rhabdovirus has the typical shape. It's barrette-like virus particle, and rabies virus belonging to the genus Lissa virus. Lissa means mad dog in Greece language. And rabies virus is classified Lissa virus genotype 1, and genotype 2 Lagos bat Lissa virus, genotype 3 Mokola virus, genotype 4 Duvenhaig virus, genotype 5 and 6 it's European bat Lissa virus type 1 and type 2, genotype 7 Australian Lissa virus. And recently, the many unclassified new genotype Lissa virus were isolated in the world. Alaban virus, Irkut virus, Fujian virus, West Caucasian virus, and so on. This slide shows the presence of rabies. Orange color countries indicate presence of rabies, and green color Australia, some country of Europe is indicate rabies free in only terrestrial animals, and white color country Japan, New Zealand, and Nordic countries, and some of European countries indicate rabies free in terrestrial animal and bats. This is a prevalence of rabies. This map was constructed by Ministry of Health in Japan government, Japanese government, and orange countries in the presence of rabies, and especially orange countries with lines, Asian countries, many Asian countries, and African countries. More than 100 people die annually. And blue country, Australia, New Zealand, Japan, United Kingdom, and Nordic country is rabies free. This means rabies free in terrestrial animal. This is a distribution of risk levels for humans contacting of rabies 2013. Green color countries this is a high risk countries, and next moderate risk, and next green color is low risk, and white region is no risk. A few countries and a few islands only. So you understand rabies and other rabies-related risk virus exists in the world, and this slide shows the distribution of risk virus, including rabies virus-mediated animals, the rabies vector and rabies transmitter. Many rabies vector and transmitter animals in the world. So prevalence of rabies has the epidemiological feature based on distribution and ecology of each host animals. For example, in your country, in the United States, many wildlife animals And raccoon rabies distributed in the mainly East Coast regions and little it's a Texas state, and skunk rabies, skunk rabies located in East Coast and the South Central region, and others. And fox rabies is also located in the East Coast and Texas and Arizona. I'm not sure. And interestingly, the bat rabies distributed in the almost all states in United States, because bat species distributed all of states. This graph shows the cases of rabies among wildlife in the United States by year and species. For 30 years, green line indicate fox, and the red line indicate raccoon rabies. And the peak is 1993 and gradually decrease. And purple line is skunk, and blue line indicates the bat rabies, a little gradually increase. This picture shows the dog rabies and cat rabies in United States 2014. Dog rabies case 59 and cat rabies a relatively higher number, 272 cases. And cat rabies distributed in the East Coast region. This distribution pattern is resembled to the raccoon rabies and skunk rabies. And dog population and cat population do not maintain rabies virus by themselves. Temporarily, the rabies virus from wildlife to domestic dog and domestic cat. So, as your country, transmission cycle of rabies is very complicated. The wildlife rabies, the carnivore wildlife rabies, bat rabies, and sometimes dog and cat rabies. So this is a question: which animal is infectious source for human beings? Fox, raccoon, skunk, cat, dog, or bat? To solve this question, one of the answer is molecular epidemiology. This is a rabies virus particle. We can extract the rabies virus genome. Rabies virus genome is RNA. This is a structure of rabies virus genome: nucleoprotein, phosphoprotein, matrix protein, glycoprotein, and large protein. 5 structure protein has. We usually will target gene for nucleoprotein or glycoprotein. We amplified part of nucleoprotein gene or glycoprotein gene, and we analyze, we determine the sequence of rabies virus genome, a part of rabies virus genome sequence, and we can construct a phylogenetic tree by using genetic distance, genetic substitutions. So we can get a phylogenetic tree, this dendritic tree like this. Here, this phylogenetic tree is this large cluster constructed by bat-derived rabies virus isolates. And upper clusters constructed from dog rabies virus isolate and fox rabies virus isolate and skunk rabies virus isolate and raccoon rabies virus isolate. So you can see each cluster, each group depend on a host animal species. This phylogenetic tree is list of viruses, including genotype 1 rabies virus. Here, number 1 group is rabies virus group. This is included in the phylo group 1 and phylo group 2, phylo group 3 or 4. You can see the rabies virus distribution is worldwide, and the host animal is carnivore and bats, and sometimes transmit the rabies virus to human being from carnivore and bats. And another list of virus, number 2 to number 15, it's new genotype of list of virus. Its distribution is Eurasia, Australia, Africa. And the host animal is only bat. It's limited. So maybe the rabies virus is maybe evolve in bat population and evolve to adapt to carnivore and worldwide, spread worldwide. And here, if rabies virus vaccine used for another risk virus is effective or not effective. Phylo group 1 risk virus is effective. Some of them partially. But phylo group 2 and phylo group 3 or 4 phylo group is not effective because a distance from the rabies virus genetically and antigenetically. This slide shows Lissa virus is isolated from only bats, not carnivore. Interestingly, rabies virus in bats distributed only American continent. And the other continent, other Lissa virus is distributed. ABLV in the Australia, EBLV1 and 2 in Europe, and Lagos virus and Duvenhage virus located in Africa. A new type of this virus, Araban Kujan, Irkut, West Caucasian, Bokero, Lirida bat virus, Ikoma, Shimoni bat virus, located in Eurasia, Europe, and Africa. From here, I would like to introduce my representative work, Molecular Epidemiology of Rabies in Brazil. This is a collaborator in Brazilian person and Japanese collaborator. Around 20 years ago, we met very great collaborator, Professor Fumio Homa Ito, University of São Paulo, and Professor Adolata Carvalho, University of São Paulo State University Paulista. We can collaborate work using the many field rabies virus sample. As in United States, Brazil has a lot of wildlife, and in addition, Brazil has a very special type of bat, vampire bat. So the transmission cycle of rabies in Brazil is so complicated. This is a vampire bat. Now he lick blood from a bird, and these animals is victim of vampire bat, cattle, horse, and pig. Sometimes rabies virus transmit from vampire bat to livestock animals, and occasionally human beings. This is a victims by rabies in prevalence region in Brazil: pig, cattle. This is a special type of the zebu species cattle and cat. And unfortunately, this girl was a victim of vampire bat related rabies virus. And the veterinary point of view, this vampire bat is a victim of rabies. This is a phylogenetic tree of Brazilian rabies virus isolate. We can divide two main groups: carnivora rabies virus isolate and Chiropteran bat rabies species, bat rabies virus isolate. And carnivora group has fox type and dog type, and bat group has insectivorous bat and vampire bat and frugivorous bat rabies virus isolate. Here I wanted to introduce vampire bats in Latin America. Only 3 species of vampire bats exist in the world, only in Latin America: Desmodus rotundus, Diphyra caudata, Diemos youngi. This Desmodus rotundus species is common vampire bat. Its population is very big, and other 2 species is not so many. This is a common vampire bat, Desmodus rotundus, distributed in the only Latin America, only in up to 2,000 meter sea level. And vampire bat has characteristic teeth like this, and this is a vampire bat bite in the foot thumb in human. It's a typical bite region. This is, I show you the video clip of the vampire bat breeding. It's a little artificial. Now he is seeking the point of breeding. Now he cut here and lick the blood from this cattle. I took this photograph in the Brazilian cave. This is all bat is vampire, common vampire bat, Desmodus rotundus. Desmodus rotundus make a colony, not so big colony. It's 100 to 100 to 200 individuals, and vampire bat not long migratory species. It's a settling species. And this is a very crowded vampire bat. All bats is Desmodus rotundus. Among vampire bats, individuals transmit rabies each other. This is also common vampire bat. He has wound in the face. This is a route of transmission of rabies. This cat is a rabies victim in the Maranhão state in Brazil. Maranhão state located in the northeast region here. This cat shows vomiting and don't like light. And this symptom is photophobia. It's a dehydration. And final stage, lymph paralysis, and very characteristic crying voice. The people took this cat for rabies diagnosis to laboratory, but during the transportation, this cat died. So cut the head and the body is burned out in the field. And for diagnosis, this cat was rabies positive. And after molecular epidemiological investigation, this cat rabies identified vampire bat transmitted rabies. This publication, our works, molecular epidemiology of Brazilian bat rabies. I will show you some of them. This phylogenetic tree indicates bat rabies virus in Brazil, or rabies virus sample from bat brain. You can see the big cluster, cluster 1 is constructed by vampire bat rabies virus isolate and frugivorous bat rabies virus isolate. indicate a green underlying sample is the frugivorous bat rabies virus isolate. So we speculate this cluster is maintained between vampire bat and frugivorous bat each other. And other clusters, cluster 2 to cluster 9, it are constructed by insectivorous bat rabies virus isolate. You can see many clustered cluster 2, cluster 3, 4, 5, 6, 7, 8, 9. This cluster group depend on host bat species. In Brazil, a lot of species of insectivorous bat, so the rabies virus is also big diversity. This is a typical frugivorous bat in Brazil, in Latin America. Altuvius retroatus is located in the Latin America. This distribution, the overlap with distribution of the common vampire bat. So maybe each other transmit rabies each other, frugivorous bat and vampire bat. And this photograph, I took this photo after eating by the Altuvius. I found this fruit in parking lot in University of São Paulo. This is a very urban area, and I pick up a seed. It's also in urban area in São Paulo city. So there is a risk of rabies infection from Altuvius in Brazil usually. Here I have to explain frugivorous bat species. This is a I showed Altuvius retroatus in Brazil, in Latin America. This species classified in Microchiroptera as small bat, and the other hand, in Africa, Asia, Australian frugivorous bat is classified in Megachiroptera, the big species, look like a fox, flying fox. Some people call it flying fox. It's not the same species. This is this Latin American frugivorous bat is Microchiroptera. And we found an interesting rabies virus isolate, Rabies rabies virus. ありがとうございます。すいません。This bat species is migratory species, and we found the Brazilian Rabies bat has rabies virus. This red isolate, red 3 isolate, and some Rabies derived rabies virus isolate located in the same cluster. So we speculate this Rabies derived rabies virus transport from North America to South America. is very the migratory rabies virus isolate. And we found vampire bat transmitted cattle rabies in Brazil. We could get a lot of cattle rabies virus isolate, and we analyze these isolate. So more than 99.2% is cattle rabies derived from vampire bat variants. So the almost cattle rabies is transmitted by the vampire bat. And we found a lot of small clusters in vampire bat variant cluster. We plot these isolate in Brazilian map. We can find some regional distribution of cattle rabies, vampire bat transmitted cattle rabies in Brazil. The rabies viruses might be circulated among bat population, vampire bat population inhabiting in contiguous areas, and natural barriers such as mountain range or river may play an important role in delimiting rabies foci. And other research work is evolutionary history of dog rabies in Brazil, and rabies virus transmit from companion animal to wildlife animal. This picture shows not domestic cat. This is a captive wild cat in North Brazil. This is illegal. And this is not a domestic dog. This is a captive fox, captive wild fox in North Brazil. This cat has a collar, and this cat has a chain, like a dog and domestic dog and cat. We have the hypothesis of expansion of dog rabies from Europe to European colonies, New World. Many years ago, people immigrant to the New World. Along with human being, domestic dog go to New World. Along with dog rabies go to New World. This phylogenetic tree is constructed by only carnivore rabies virus isolate in the world. Cluster 1 and cluster green color, cluster 1, orange color is cluster 2, is constructed by domestic dog rabies and domestic cat rabies virus, and cluster 2 is constructed by domestic dog and wild foxes. This is the Brazilian carnivore rabies clusters. The outer branch, this clusters constructed by European dog and European fox rabies virus, and North America, Mexican and carnivore, and Middle East and African dog, domestic dog virus clusters. Fox rabies is very interesting. However, it is difficult to get the wild fox samples. So we use the roadkill sample, like this photograph, and we analyze only brain sample. And this slide shows this green cluster and orange cluster is constructed Brazilian carnivore rabies virus isolate, and outer cluster is constructed European dog rabies virus and European colonies countries rabies virus isolate. So if we find when this node A, node B, branch A, branch B is the estimated age or year, we understand transfer age from European country to Brazil. So we calculate the estimated branch age, node A and node B. This, as a result, node A is 1878 to 1904. Node B is performed 1909 to 1935. So we think during late 19th century and early 20th century, domestic dog transfer rabies virus from Europe to Brazil, and after that the rabies virus had spread over the area in Brazil, and then interspecies rabies virus transmission between dogs and fox had occurred. This is the last question. Does risovirus exist in Japan? Of course, no rabies viruses naturally in Japan since 1957. But in Japan, bats that risoviruses are isolated in other areas and their closely related bat species are inhabited. For example, an emerging risovirus, West Caucasian bat risovirus, or its related virus may be widely distributed in host bat from Eurasia to the African continent. This is a very newly report. Novel bat risovirus were discovered in Taiwan. This new bat risovirus isolated from Japanese house bat. This bat species distributed Taiwan to north areas of Japan. So this species, bat species, is distributed in Japan. This two newly bat risovirus is located in the phylo group 1 in the dendritic tree. So this risovirus is included in phylo group 1, but identity, nucleotide identity is 63 to 75%. This score is not so high, so this risovirus is maybe new genotype. So if we perform the very active surveillance in Japanese bat, we may discover new risovirus in Japan. This is a list of new risovirus in Taiwanese. Many unknown factors, zoonotic or not, is unknown. Human case, not yet. Incubation time in humans, not unknown. Case fatality rate in human is unknown. Human being symptom, unknown. This is very new risk virus in Taiwan. Oh, okay. This is the last slide. This is our research supported by the many collaborators, Brazilian side and Japanese side. Professor, Emeritus Professor Fumio Honma Ito, he's a great collaborator in University of São Paulo, and Professor Adorata Carvalho is also the good collaborator in University São Paulo State Paulista, and Japanese side, Professor Takeo Sakai, he was my boss, and he was president of Nihon University. And Dr. Yuki Kobayashi, she is a good collaborator. She is working in Nihon University as assistant professor, and many graduate students now they are working in institute or some prefecture sections. And we research, we can research supported by many research grants from Japanese government. Okay, thank you so much for your attention, and I appreciate for having this lecture. Thank you so much.
Okay, anybody have any questions?
Hi, Captain Kearney. I've got a few questions that came up. One question is, so you're doing a phylogenetic analysis on the genome of the virus, right? How much difference in the sequence do you need in order for it to have that branch off into a separate group? Because you said that in order for it to be a new virus, it was 62.9 to 75.1% different to get that big branch off. How fine of a difference do you need to get those different sub?
New classification?
Within this, so those deepest down ones, how much of a difference do you need to get that difference?
Maybe to divide the list virus, list virus classification is maybe 70 to 80%. But rabies virus, in rabies virus, maybe the totally I think the 19% is the same.
Okay.
And depend on the length of the analysis region. If you use the very short fragment, it's difficult to find out. But if you use the full genome, 11 kilobase, it's a very good difference. Yeah.
I guess as a quick follow-on to that, have you done an analysis of where those differences are in the genome? Is there a change in the amino acid sequence, or is it just purely the nucleotides that are changing? Because as you go species to species, based on the replication in the host cells, you could have more of a propensity to get a certain nucleotide change that will still have the same effect on the amino acid. So have you done any kind of analysis on the amino acid sequence group?
Yeah, I think in the I showed you the Uh, this shows this slide shows genetically distance, and maybe the amino acid change is significant between this group to another group.
Right, because
Yeah.
you did mention that as they're further apart, the immunoglobulins they can use don't really work on the other ones. So that's indicative of
Yeah,
either
yeah.
the glycolipids
Yes,
are
yeah.
changing or the amino acids are changing, the
Mm.
sugars are changing. Yeah. So, okay, thank you.
So, but I think the sometimes, as you know, the survival rabies case in United States, some maybe 2 or 3% survived.
Right.
It's that was might be abortive infection.
Mm-hmm.
It's maybe that abortive infection from bat rabies virus. So I think the might be the amino acid change to another immunoreaction. Yeah. Yeah.
Thank you.
Kafan here. Well, thank you for a wonderful talk. I just have a kind of similar question. Is so as not as we all so basically by your phylogenetic tree, it looks like a virus origin from bats, then jump to species. So have anybody looked into the whole genome comparison to see which it's a very compact virus, which protein mutation caused the species jump? I mean, a lot of time it happens in the glycoprotein because that's how you gain access to the cell entry. So have anybody who looked at it, the difference between a bat and dog and human to see which one actually changed, get educated guess?
Mainly the researchers showed the glycoprotein gene, yeah. But some researcher investigate another gene, nucleoprotein and large protein. It's polymerase. It's called polymerase. So some researchers investigate some part of the large protein polymerase gene now.
Oh, okay.
It's very good.
I have a second question. So you showed the immunostain. You showed that a lot of viral particles in the central nervous system. So, but also saliva is also a route of transmission. So, have anybody who did similar staining to see which cell type actually virus affect? And have anybody who had done PCR on the saliva to do diagnosis of the virus?
Yeah, yeah. But the RT-PCR detection is some of failure. Especially the rabies virus in saliva, it's yesterday detection and today not detect. So it's not established technique. Of course, RT-PCR has a good sensitive diagnostic technique, but sometimes the failure.
So another question is anybody who have done immunostaining on other cell types to see what kind of the virus affect other cells or not?
Sorry,
Oh, sorry.
I can't understand.
Have anybody who have done immunostaining on other sites, cell type other than central nervous system?
Other
than-
side.
Yeah.
Interesting research. Some researcher detect the rabies antigen from the here.
The hair follicles. They've
Yeah.
done it. They found them in hair follicles, but like you said, is you have a lot of false negative. So it's not a gold standard yet. That's why they keep going back.
I'm just interested because it looks like it mainly affect the brain, but you also have viral particle in the saliva, so there must be some pathway that
what is it, the fifth cranial nerve is what they say it kind of comes from the brain down the fifth cranial nerve that feeds the mouth and everything. But yeah, and that's the other thing too is why it's just kind of intermittent. It's not always in the saliva. It's just there. Different line numbers.
Thank you.
Sir, okay. David, your distribution from the phylogenetic groups, how do they compare to the rural and urbanization of Brazil? Do you have like a higher prevalence of certain clusters in more urban areas than you do rural areas, and vice versa?
それいいですか。もっとたくさん調べないとわからないですが、吸血コウモリの狂犬病ウイルスは都市部とそれ以外の場所で分布が違います。そうですね。はい。ヴァンパイアバットは都市部にはいない。はい。でフルーツバットは都市部にもたくさんいます。
He said that they need more research, larger number of research. There's a difference in the distribution of the fruit bat and vampire bats, and that kind of reflects. So vampire bats, they don't see much in the urban area, so that particular holiday categories are not distributed in the urban. Fruit bat, they go to the urban communities and they see that type of that particular philo group in urban area. So it's a, I think it's a matter of the distribution of that particular members, the carriers of that particular philo. Did you answer to you?
Yeah, of course. Well, that they need more data, yes.
They need more data.
Thank you. I just assume that what you see in the more insectivorous bats would be in the more rural area. What you saw in like the carnivores may occur more in an urban area because of, more domestication with dogs and stuff. And so I was just curious if there was any indicators of that. But it seems to me like it would be more
If it's reflected to the philo group,
yes.
で、そのファイルグループにそういうディストリビューションの違いっていうのは出てきているってこと
はい。
ですね。Yeah, I think yes. Yeah. This is difference in the philo group in the urban area and the rural area.
That's mostly dependent on the host animals.
So that one that appears to be the research of things larger group of research.
Okay, Rabies Presentation, do you have anything to say for this class?
Well, thank you for coming out and giving a presentation to everybody here from at least from Public Health Activity Japan. I'd like to give you one of our coins. The PHA's Public Health Activity Japan, and then on the back we do have the vet caduceus and some animals, of course, One Health and everything. So thank you.
Thank you so much.
Yes.
Yes.
Thank you.
Thank you so much.